just going to pick up where we left off the other day. It's just a recap because I think we finished a little bit past here. But this, um, what we just described was transformation, and specifically bacterial transformation. So we discussed the experiment that Griffith did to prove the theory of transformation and the breakthrough that it led to associated with the transformation of genes from one strain of bacteria to another. So we talked about that experiment, and it led to this amazing breakthrough that genes had to be the transforming factor, right? Because a quick recap, that when he mixed the harmless bacteria strain with the heat-killed bacteria strain, it actually caused the mice to develop pneumonia and die. And when they extracted the strain from the dead mouse, they found the live disease-causing bacteria strain. So transformation had to have occurred between the heat-killed bacteria and the harmless bacteria. Okay, so a group of scientists wanted to determine which molecule in the heat-killed bacteria was most important for transformation. So what they did is they destroyed the DNA in the mixture. And when they did this, the transformation didn't occur. So what that meant was that DNA had to be the transforming factor. And even more so, that genes had to be located on DNA, because we knew that the gene that produced pneumonia was being transferred, but we didn't know which molecule it was on. So this experiment proved that genes were located on DNA, and DNA was the thing that was being transferred to the harmless bacteria strain. So the next thing that we're going to talk about, and it's actually incredibly relevant right now, is bacterial viruses. And several different scientists repeated the experiments on bacteria, but it was Alfred Hershey and Martha Chase who performed the most important of the experiments relating to this discovery. So this is what I wanted you guys to read in 12.1. I wanted you to focus on the experiment that they did, because we're going to talk about it, and we're going to see why it was so important and what it meant. So Hershey and Chase studied viruses, and viruses are non-living particles that can infect living cells. That's really important because a lot of people just consider viruses as living things, but they're not. They can infect living cells, they can move around, but they cannot replicate on their own, meaning they can't divide without invading and infecting a host. So therefore they don't satisfy the cell theory, and therefore they're not considered living things. So what we're going to talk about is bacteriophages. And a bacteriophage is a virus that infects a bacteria. Okay, it's not a type of bacteria. Again, viruses are non-living. So a bacteriophage is bacteria that has been infected by a virus. So the kind of virus that infects bacteria is known as a bacteriophage, which means bacteria eater. And a typical bacteriophage is here. So you can see this is a T4 bacteriophage. So this is actually a really advanced bacteriophage, and it's really effective. So you can see that it holds its DNA in its head, right? And then you can see the tail sheath, okay? So what happens is you can also see the tail fiber. So what happens when the virus goes to infect bacteria is those tail fibers will grab onto the cell membrane of the bacteria, and then the tail sheath will implant itself through the cell membrane, and its DNA will shoot through the bottom of it into the cell. And then, since bacteria is prokaryotic, meaning it doesn't have a nucleus, it's very easy for that DNA to implant itself right into the bacteria's DNA. And that way, when the bacteria divides, the viral DNA just kind of hops on for the ride, and it will replicate as well. Um, what the viral DNA actually says is to make more viruses. So when the bacteria is just carrying out its day-to-day operations, a protein might come across that DNA strand that the virus implanted and read it and actually manufacture more viral particles. So the virus will multiply and multiply. So it will actually make more viruses within the bacteria. And once the bacteria has divided enough times, eventually those viruses, those new bacteriophages, will burst out of the bacteria and go and infect more bacteria. So it's actually a very advanced and effective method. So this is kind of what I was just saying. When a bacteriophage enters a bacterium, it attaches to the surface of the bacteria cell and injects its genetic information into it. The viral genes produce many new bacteriophages, which destroy the bacterium. And when the cell splits open, hundreds of new viruses burst out. Then the Hershey-Chase experiment is the next thing that I want to talk about. So the scientists Alfred Hershey and Martha Chase, they were studying bacteriophages that were composed of a DNA core and a protein coat, very similar to that bacteriophage. They wanted to determine which part of the virus, the protein coat or the DNA coat, entered the bacteria cell, because at that point they didn't know what it was exactly, as far as viruses that made them produce more viruses. They didn't know if it was the protein coat that was in charge of making more viruses, or if it was the DNA. So Hershey and Chase grew viruses in cultures containing radioactive isotopes. So they had some viruses that had phosphorus 32 and some that had sulfur 35. And the radioactive isotopes don't actually affect the viruses at all. It just makes them it makes us able to see which is which. So it's basically like a tracker. So since proteins contain almost no phosphorus and DNA contains no sulfur, these radioactive substances could be used as markers to tell which molecules entered the bacteria and carried the genetic information of the virus. So if they found radioactivity from sulfur 35 in the bacteria, it would mean that the virus's protein coat had been injected into the bacteria. Whereas if they found phosphorus 32, then the DNA core had been injected. So you can see in the image that we have some viruses with phosphorus 32, and you can see that the DNA in the head there is all coated in phosphorus 32. Then you can see the bacteriophage with sulfur 35, and that's all around. That's not touching the DNA at all. That's just coating the protein around it. Okay. So they take these viruses, they mix them up with bacteria cells. They allow the viruses to inject themselves into the bacteria, and then they take a look at the bacteria. And what they saw was that they found phosphorus 32 inside the bacteria, and they didn't find any sulfur. So what that said is that since the DNA was marked with the phosphorus 32, that phosphorus and the phosphorus 32 was now inside the bacterium, that meant that the DNA was the substance that the viruses use to infect the bacteria, and the protein coat was not involved. So nearly all the radioactivity in the bacteria was from phosphorus, the marker found in DNA, and Hershey and Chase concluded that the genetic material of the bacteriophage was DNA and it was not protein. So the role of DNA, we're going to talk about this going forward with the structure of DNA first, but the DNA makes up genes. That must be capable of storing, copying, and transmitting the genetic information in a cell. And these three functions are similar to the way in which you might share a treasured book. I think of DNA as like a big cookbook, and it's full of recipes. Now, every single cell has the whole cookbook, but they might only use certain recipes depending on what they're trying to make. Okay, so the primary job, though, of DNA as a molecule of heredity is just to store information. And genes control patterns of development, which means that the instructions that cause a single cell to develop into a complex organism must somehow be written into the DNA, and it is. And that's what we found, is that all of your genes are on DNA, and your genes provide all the instructions on how to make you you and how to keep you alive. So before a cell divides, it must make a complete copy of every one of its genes. We know that this happens in the S phase of interphase, and it's similar to the way that a book is copied. So, to many scientists, the most puzzling aspect of DNA was how it could be copied. And once the structure of DNA was discovered, a copying mechanism for the genetic material made a lot more sense. Because before, we didn't know what DNA looked like, but as soon as we figured out that double helix structure, then we clearly understood how DNA replicated itself. And that's kind of where we are going to leave off for today, and we're going to pick up on next class. This is all just meiosis stuff. So guys, we've covered this. If you are still a little bit unsure of meiosis and mitosis, please read up on that or let me know. And there's some stuff that maybe I can provide you with that can help you practice remembering some of the different phases of mitosis and meiosis and what happens at each of those phases. Okay, so what I want you to do is submit the notes, the guided notes for this into the assignment folder. Okay.